Hello Biotechnicans. This is Dr. Farhan Zamir from Biotechnica, Bangalore. So from few days, all our interns and most of our PhD science enthusiasts, they were requesting on a specialized video, and this is mainly on microbiome research. Now, the word microbiome has become the buzzword, and everybody wants to have their research perspectives in terms of microbiome. Now, let us try to see why is this making such a big noise. Let's dwell in. So, welcome back. When we talk about microbiome, first we need to understand what is this microbiome. See, remember, we are just a universe of microorganisms. So our body is made up of tons and tons of microorganisms which will define life. So what I am is what the microbiome, what I contain. So it becomes very, very crucial the kind of consortium of microorganisms what I carry. So in scientific research, what people are trying to do is all clinicians, all scientists, all researchers are just with a microbiome research because they are trying to find an alignment, a trajectory marker towards how exactly I can define a disease in terms of microbiome so that this can be a potent biological marker or a diagnostic marker. So when I talk about microbiome, it is highly specific. Let me take you an example and then try to explain. Example, if I take my fingerprint, now my fingerprint matches 10 people in the world. Or if I take my iris, the pattern of the iris matches 3 people in the world. However, the microorganisms which are present in my gut, that means to say the gut microorganism even does not match with a Siamese twin if I have so, and it is what I'm trying to make you understand is that it is that highly unique, and this contains very specialized character. So what people are trying to do is they are trying to dwell an interaction or an association of the living system with that of your habit and habitat so that these could be used as a potent biomarkers for diseases and therapeutics. Now, when I talk about microbiome, it is mainly the gut microbiome. So the gut microbiome could be associated with the physiology of a living organism. It can be associated with the growth of a living organism, then its habitat, then very importantly, the tolerance to test. The stress tolerance has been tested using a gut microbiome, because even if I don't sleep well, the consortium of microorganisms is getting differed. If I don't eat well, again the consortium is getting differed. And if I am diseased, again there's a new set of microorganisms. So what people are trying to do is looking into the stress biomarkers, people are trying to associate that with dysbiosis or eubiosis of microbiome. Not to forget, again the gut microbiome is associated with the immune system. It can actually directly have an impact on the B cells and T cell population and their subpopulation, and very importantly, dysbiosis can directly create diseases and disorders. So with this as an holistic approach, everybody at one point or the other, they're trying to understand gut microbiome or the microbiome of any kind of a particular organ. So how are these microbiome studies done? In the current scenario, there are 5 different methodologies in which microbiome research has been done or carried out. Now, the first methodology is the metagenomic microbiome profiling. The second one is multivariant microbiome analysis. The third one is diet, microbiota and host interaction. And the fourth one is interaction between the microbiota and the host immune system. And then the fifth one is the microbiota with the targeted interventions. Either it could be a phytochemical interventions or it could be some chemically synthesized interventions so that a potent drug therapy comes out of this particular kind of research. So these are the 5 different types of niche areas wherein microbiome research is on a high boom. Now, let us try to understand how exactly this has been done. If you can just imagine that I want to look into a microbiome on my skin. So I am now not talking about the entire microbiome which is making me, that is life, but however, I am talking about a set of microorganisms which are residing on my skin. So hence we call them as skin microflora or skin microbiome. So when I talk about microbiome, I am talking about the entire consortium, entire group of species and phyla of microorganisms which are actually getting established on my skin and which will define my life, that in particular which will define my skin and its microbiota or its richness. Let us try to see into this. So normally, to identify which are the microorganisms which are present on a given organ, in this case it is the skin, there are two different methodologies. The first methodology is the Amplicon method, wherein we are trying to look into the amplifying biomarkers using universal primers, or the second methodology is to look into the whole genome metagenomics. So whole genome metagenomics will give you an entire profiling of microorganisms which have been present. However, coming on to the first type, which is the Amplicon method, in Amplicon method, we are trying to design universal biomarkers, very importantly, for understanding the bacterial consortiums or for understanding the fungal consortiums. So, for bacterial consortiums, we normally use 16S rRNA studies, or for fungal analysis, we take up, ITS regions. ITS1 or ITS2 regions could be used for studying 18S, 5.8S, and then 28S ITS regions. However, in whole genome metagenomics, we would take the entire fragment of the DNA, which could be from either bacteria, fungi, viruses, protozoans, or even host, that is the eukaryotic microorganism, eukaryotic species. So here, what we do is we sequence it either on an HiSeq, MiSeq, or Illumina platform. And once we do this, then using various kinds of approaches, that is various kinds of tools, and one such beautiful tool which could be used is R, for microbiome research, it is called as Chime, and also another beautiful tool, an open source tool, is called as Mother. I think we have also made a video on the various kinds of data analysis, especially for big data analysis using Mother and Chime. So once I do this particular Amplicon data, then I will be able to understand the richness and diversity of microorganisms present in my particular sample. However, if I go for the whole genomic metagenomic approach, then I'll be able to give not only the kingdom, but I'll be able to give up the phyla, and also very importantly the species variation which is actually present in the given system. So this is how, in a nutshell, we try to analyze the microbiome data, taking skin as an example. So now the entire factory of microorganisms starts with the gut, that is the intestinal microflora, and understanding this intestinal microflora and its trafficking becomes very, very crucial for microbiome research. Now, if you look into the intestinal microflora, it is containing an organism diversity and richness of around 10 to the power of 14, that is more than 500 species. They reside in only the gut, and hence, understanding the entire interaction in the pure culture level, the mixed culture level, and then in understanding the host pathogen interaction becomes very, very important. So in this particular picture, you are able to see that a special species of Lactobacillus lactis, Lactobacillus cremoris, Streptococci, Staphylococci, E. coli, then Fecalis, Clostridium, Chlamydia, all these microorganisms, they are always in interaction to define life. So let us try to see that what are the factors which actually affect microbiome. The first factor is the environmental factors, which could be physical or physiologically, which can affect the microbiome. And then, because we have been prone for various kinds of antigens, we always pop in the defense mechanism drugs, which are called as antibiotics. So this antibiotics can also have a huge impact on the individual microbiome. Then the bile acids and the concentration of bile acids can also play an important role. And then these microorganisms which are already residing in a given system, they produce a special kind of peptides which are called as antimicrobial peptides, and these antimicrobial peptides can also play an important role. Then coming on to the gaseous exchange, the quantum of oxygen and quantum of carbon dioxide in your system can also define the quality and quantity of microorganisms which are present in your system. And then very importantly, the host genetics the host diet, and whether the maternal factors and paternal factors can also play an important role, and whether an individual, when he was or she was as a fetus or as a baby, what kind of microflora was the fetus exposed, or when it has been born, whether the baby was born in terms of C-section or it was through a normal labor, or whether there was a pet in the house when the baby was brought into the house, also makes a difference in the change in microbiome. So what I'm trying to make you understand is this interaction of host to specific interaction with the microbiome and the habitat defines a living system. So even whether the baby was breastfed or whether it was with formula milk also makes a huge difference, and what was the host immune response of an individual can play a predominant role on the entire consortium of microorganisms. So these are the various factors which can define a microbiome in a given individual. Now, as I was trying to tell you that these microbiomes or these microbiota can actually define health, or it can actually define a disordered condition or a dysbiosed condition. So if you see all these factors which are in green in color, so this will define health. So example, higher concentrations or higher quantum of short chain fatty acids, higher concentration of antioxidants, then improved lipid metabolism, low gut inflammation, insulin sensitivity, all these can actually reduce the risk of infection. So in a given system, if there is a higher fiber intake, higher probiotic intake, this can actually define a good health of a given organism. On the contrary, if I have lesser concentration of short chain fatty acids, if I have higher concentration of LPS, if I have an increased CVD risk, if I have a gut inflammation, epithelial inflammation, if I have insulin resistance, all these factors, including the change in the pH, can also play a predominant role in the habit and habitat of the microorganisms which are residing inside a system, and this can in turn reflect the health of a given individual. So these are the biochemical factors which actually defines the resident microorganism in a given system. Let me try to take an example. Let us talk about the skin microbiome. So if I want to study a skin microbiome of a given individual, let me try to divide the skin into three different compartments. The first compartment, we will just call it for the sake of our discussion. The first compartment would be the moist area, something which is under the armpits or beneath the thighs. So here this area would be the moist area. Or I can still consider another area which could be a dry area, which have a direct access to sunlight, and wherein this can lead into the dermal drying. Or I can have the sebaceous sites, wherein these sebaceous sites could also create a different kind of microflora. Now the important point what I want to really make out is, if you look into the moist site, dry sites and sebaceous sites, the kind of microflora and the CFU, that is the colony forming units, we normally in terms of microbiome research we call it as OTUs, that is operational taxonomical units. These operational taxonomical units actually differ on the same site, okay, with three different areas on the same organ can also differ, and skin is a classical example wherein you can have higher concentrations of one microorganism at a particular site, then the same microorganism can get lowered on a particular site on the same organ. So here the skin microbiome can show a large kind of diversity. So continuing the discussion, I can still have, now from the skin, I can go into the oral microbiome. Now the oral microbiome also plays a very, very important role, and hence this can have a consortium of organisms such as fungi, I can have viruses, bacteria and archaeas. So all these put together, which are dependent on various factors such as age, host and the environment, habitat, and also very importantly the biofilm formation or the biofilm forming ability of microorganisms, which could be either tested by crystal violet staining method or ruthenium red staining method to understand the strength of the biofilm, or also to understand the hexapolysaccharide material which has been excreted by these microorganisms. Just for an analogy, when you get up early in the morning and before you brushing your teeth, if you swirl your tongue on your teeth, there is a slimy layer. This slimy layer is nothing but biofilm which have been formed by the friendly microorganisms, and because of this you do not suffer with a disease. By any point of time, the same biofilm is being produced by a pathogenic variety of microorganisms. This can lead into dental caries, and it has also been seen that it is published in a beautiful paper which was published in Nature, wherein the dental caries, the microorganisms from the dental caries can travel into the bloodstream and cause vascular disorders. So, it is now seen that certain microorganisms, they are not just restricted to particular site, they are also able to navigate from one site to another site, and these are called as access. Now, these oral microorganisms or buccal cell microorganisms or the mouth microbiota can also define the oral health of an individual, whether the person could this can act as a diagnostic marker for oral cancer. Now, as I was trying to tell you, that these microorganisms can navigate from the site of their flourishment and to another site of navigation, and these are also very important in defining obesity, diabetes, liver disorder, pancreatic cancer, colon cancer. All these kind of systemic diseases could be defined from oral microbiome. So what people are trying to understand is, how do I use this oral microbiota as a diagnostic marker for all kinds of lifestyle disorders? Now, again, it is not just obesity and few disorders. Gut microbiota could actually get associated with the brain, with the thyroid, with the muscle and the bone, then very importantly, the inflammatory disorders, and then obesity and cardiovascular disorders. So, you name a particular disease to which, we can still get microbiome associated, and then analyze this at a molecular or sub-molecular levels. Let me take another beautiful example of vaginal microbiome. So, when a female baby is being born, so from the birth of the female baby till the adulthood or till aging, I can actually see a varied kind of microbiome consortium which will get differed. So, if you look into this particular, the richness or diversity. So in childhood, when the baby is born, if I just take an example of lactobacillus, so these lactobacillus are in a medium concentration. But however, when it comes to the reproductive age, the same lactobacillus actually increases in number. But however, towards menopause, the same lactobacillus again goes on to very lower concentration. So now, understanding the consortium of consortium of microorganisms, especially bacteria, with special reference to lactobacillus, can define at what stage of your life are you in, especially in terms of your reproductive cycle. Let us try to understand this in a much better way, wherein these vaginal microbiome can also indicate the hormonal status of a given individual or a female. So these are the hormonal status in an adolescent. Then these are the hormonal status of an adult woman. So in an adult woman, you see a predominant lactobacilli species, and again, whether the female is on her periods, or whether the female is has conceived, or whether she is on her pregnant cycle. So if we are able to understand the microbiome, these microbiomes are very, very specific onto a particular duration in a particular individual. And then when I look into the same female, when the female is attaining towards her menopause, the entire consortium actually again changes. So depending on this, we can study microbiome as a diagnostic markers. So again, as I was trying to tell you, that physiological factors such as pH can also have a predominant role. Example, you have what happens for a female when the pH is between below 6 and between 6 and 7, and when it is 7 and when it is higher than 7. So you can absolutely see that how exactly there's a variation in lactobacillus just with a change of pH of 1. Then coming on to the next example is the respiratory microbiome. even in your lungs, because I was trying to explain in the factors that oxygen also plays a very crucial role for the establishment of microorganisms, and hence in respiratory system there's a special kind of microorganism which could either require more amount of oxygen, which can even survive with little amount of oxygen, and which can survive on carbon dioxide. So depending on these, we can still understand various kinds of respiratory disorders using respiratory microbiome. Now, as I was trying to make you understand that it is not just the microorganisms are restricted to a particular site, but they are also having an ability for trafficking between one area to another area, between one organ to another organ, and this is called as the gut-brain trafficking or the microbiome trafficking. This happens with the enabling of an axis. Example, the microorganisms which have been present in my gut, either the microorganism or the metabolite which have been produced by the microorganism can travel my brain. So this is possible with an axis which is called as gut-brain axis. The same way, I have the gut-lung axis, I have the gut-skin axis, then I have the gut-liver axis, so which can actually define the health of an individual. So gut-brain axis has been largely studied for various kinds of neurodegenerative disorders such as Alzheimer's, Parkinson's, neurodegenerative disorders such as anxiety, depression, autism, and all these kinds of disorders wherein new pathways have been established with the metabolites which are getting trafficked between the blood-brain barrier. Then you have the gut-kidney axis for most of the nephritic disorders or most of the inflammatory disorders, most of the disorders such as lifestyle disorders, example, urolithiasis, nephrolithiasis, generally called as kidney stones. All this is again mediated by a set of microorganisms through a pathway, through a route, and this route is called as the gut-kidney axis. Never to forget the gut-liver axis. Again, if somebody is alcoholic, again, how exactly the microbial consortium can define the liver of the health, whether the individual is suffering with the fatty liver or whether the individual is suffering with hepatitis. So these kind of diseases could be identified with the homeostasis or with the dysbiosis of a given individual. We can still study the health of a liver. Now, coming on to the respiratory tract, also we said that you have a beautiful junction between the gut and the lungs. In case of tuberculosis, a beautiful example wherein how exactly this microorganism, that is mycobacterium, can actually affect the lung, and the microorganisms which are present in lung, the metabolites, how exactly it can affect the gut microbiota has been beautifully explained, and especially in terms of short chain fatty acids, a lot of researchers are interested with this to identify the biomarkers involved in it. Now, as I was also talking about a special kind of an axis, this special kind of an axis includes not just the gut and brain, but however, it also includes the consortium of organs such as the gut, brain, kidney. Example, I can say as HP axis, that is hypothalamus pituitary axis, wherein the microbiome is responsible for maintaining the corticosteroid levels, and then various other biomarkers, the acetylcholine esterase, butyrylcholine esterase, all these biomarkers such as acetylcholine, dopamine, serotonin, all this can get upregulated or downregulated with the metabolite or the microorganism which are present in this particular trafficking route can also define what are the various systems. So with this as a nutshell, we just wanted to tell you that these are the various kinds of research which are going in terms of microbiome. Maybe in next video, if you request, we will also try to look upon in detail that how do I actually analyze microbiome using various softwares such as Mother or Chyme, so that you can actually perform a HiSeq or MySeq in your particular research. Thank you very much for staying connected, and your success is our motivation so that we can create much more content for your betterment of science. Thank you very much.